Hello, welcome everyone to this episode 4 of our podcast series about Discovery UNESCO Global Geopark. As I said, this is the 4th episode and it's going to be on fossils. We have named this episode Fossils Frozen in Time, the Ediacaran fossils of Discovery UNESCO Global Geopark, which is pretty self-explanatory. And this episode, we're going to talk about fossil phylogeny, preservation, and ecology. So basically, the fossils found at Discovery are one of the most extraordinary fossils in the fossil record in the world. They're preserved in exquisite detail, and they reveal ancient life forms that challenge our understanding of evolution and ecology. So in this episode, we're just going to discuss key fossil discoveries, their relevance, and how they help scientists, meaning in this case us, reconstruct early ecosystems and their biology as well. And we're just going to explore why these sites are so crucial for the evolution of complex life forms. And so for this episode, we're joined by our master student, Heléna. And I'm going to let you briefly introduce yourself, Heléna. Well, thank you for coming, first of all. So introduce yourself, and we go ahead with the interview.
Yeah, so my name's Heléna. As Danny said, I'm a master's student studying with Duncan McIlroy and in Danny's lab. And I'm from Kingston, Ontario, so I'm new to Newfoundland, and it's been awesome to discover all the fossils and the sites here. So.
Great. And so the first question is, it's just going to be a discussion more than a question, I guess. But the fossils at Discovery Geopark are incredibly well preserved. Can you explain how these fossils are formed and why the preservation is so exceptional? What specific conditions allow these delicate forms, because these fossils are soft-bodied, to be preserved for over 550 million years?
Yeah. So in the case of the geopark, the Ediacaran biota lived at the bottom of the seafloor, and they're preserved with rapid burial. And usually that happens one of two ways. So we have the turbidites, which are underwater landslides. And so all of a sudden you just have a ton of sediment that just piles onto these communities. And we also have a volcanic ash or tuff, which is really fine grained, which is really nice because it preserves the really fine details. And that is another way that rapid burial can happen. So hence the episode name being Frozen in Time. They're frozen in time by this rapid sedimentation. Another thing is that there was this mat ground at the bottom of the ocean that also helped preserve the organisms. And it was really nasty, probably looking, but it helped preserve things well, which is good. And the last thing I'd like to say is that the geopark is in a craton, so it's away from tectonic plate boundaries. So even though they're really, really old, they weren't subject to subduction and lost to the rock cycle or metamorphosed to a point where they're unrecognizable as fossils anymore, which is awesome and lucky for us.
Right. Yeah, so I think, to sum it up, really, it's like two main things. One of them is like sedimentologically speaking, you have this very violent, turbid landslides, like submarine landslides, as you say, that basically smother everything and likely killing everything and destroying all the fossils at once.
Yeah, it's kind of sad.
It's sad, yeah. And so what happens is that these fossils basically get sealed, they get covered, right, by sediments, and this new layer of sediments is sealed by newly growing microbial backgrounds. We have evidence of these backgrounds in terms of like texture, organic surfaces, for example, that we call elephant skin. So it's just funny looking sediment. They also lead to the precipitation of pyrite, which we have observed. And basically what happens is that once the fossils are buried and sealed away from the water, you have sulfate reacting with decaying organic matter, and just by the action of sulfur-reducing bacteria, they form this very reduced form of sulfur that eventually reacts with iron and forms pyrite. So there's like a layer of pyrite basically on top or below the fossils, which is what we call the death mask model. So it's basically you have like a thing like that, which preserves how you look, essentially, right? So this is kind of like what happened. I mean, the model is a bit more complicated, but I think in essence is something very similar to that. Okay,
Yeah.
I think we can jump onto the second question. Okay, so Helena, the second question now. We actually didn't like that introduction. Okay, I'm gonna try again. Okay, well, very good. Now that we have discussed the preservation, we're gonna move on to the second question. So Helena, the second question is basically the Ediacaran fossils don't look like anything that we have alive today. What are some of the most iconic fossils found at the geopark, and what do we know about them?
Okay. So the first one to me as being iconic is Aspidella, which maybe not many people would say because it's not as flashy, but I think
It's
it's
not as
iconic.
flashy.
It's iconic because there are so many of them. Like if you see Ediacaran fossils, the first thing you're going to see is probably one of these like discs, these Aspidella.
Because that's what they are, essentially, just a rounded disc.
Yeah, it looks kind of like a bullseye, but that's it. Sometimes they're attached to body fossils, but most of the time they're not. We think they're for attachment purposes, but again, we're not entirely sure. There's a lot that's speculated in the Ediacaran field.
Right, because sometimes you see sometimes you see like just the discs just on their own, and sometimes you see the discs attached to kind of like a stem and then like a frondose part of the organism, right? So we believe that at least some of these guys were probably like anchor structures or holdfasts.
Yeah, I would say it was one of the more confusing things when I first studied this. Like, why are there so many of them? It's crazy to me. Anyways.
What else? What else?
Yeah. But another, like, I would say more flashy, if you will, iconic fossil at the geopark are the Rangeomorph group. And within this group, there are lots of different fossils like Fractifusus, which I know you studied a lot and study a lot, and Bradgacia as well. And this Rangeomorph group is a type of Ediacaran fossil that has a pseudo-fractal body plan. So it has these units of self-similar repeating pattern, kind of like a snowflake or a coastline. And I like to joke that this is like a discontinued body plan because we don't see that anymore. So I think that's what makes it really, really special as well. And then more recently, we have Hautia and Mumpaleobiology, which are like a big deal, at least in the scientific biology community. So, because that's the first evidence we have of muscle tissue. So that was a really, really big find that was more recent. And it's the oldest eumetazoan being kind of like the complex animal group. It's the oldest body fossil we have of that, so that's pretty neat, I think.
Right, yeah, no, it's the first evidence of preserved muscular tissue in the fossil record, as you said. I think the most beautiful specimen of Hautia, at least in my opinion, that is the most beautiful, is displayed at the rooms in St. John's here in Newfoundland.
You should go.
So
you should all go.
If you happen to be traveling by, you should certainly go to the Rooms Museum and check it out. What you said about the Rangeomorphs, this is very bizarre creatures because they kind of like, as you said, like it's their body plan is kind of like a branch within a branch within a branch within a branch. Kind of
they look like leaves, kind of.
I was gonna say they look a bit like ferns, in my opinion.
Yep.
But we know for sure these organisms were not plants. There are not many things we know for sure, but it seems to be quite certain that they were not plants because they're preserved in very deep marine sediments.
Yeah.
So basically,
no light there.
No light below the photic zone and below the wave-based action and so on. So we don't think they were capable of conducting photosynthesis or anything like that. So they must have been something else. So they were like this quilted fractal organisms with a glide.
They're so pretty.
With a glide plane of symmetry as well, which is peculiar. Like scientists have traditionally compared them to, for some, sea pens, which is just this group of octocorals, basically. They look very differently. There's lots of different types of sea pens, and they kind of look a little bit like them, but there are some differences. For example, these are the ones we have are fractal. Sea pens are not really fractal. We don't have evidence of muscular tissues in the Rangeomorphs, for example. We also don't have any of these like specialized polyps that you have in these corals.
Yeah.
So it's just that they have, I think, important differences. You mentioned Fractifusus, which we interpret as having lived reclining flat on the sediment, which would also be very different from the biology and the physiology of octocorals in general, in particular sequence.
Yeah.
So it's not very clear, I think, what they are. I think the best guess is probably like stem group Eumetasaurans, but I think the comparison to sepens is probably to be taken with a pinch of salt, I think.
Yep, it all is.
Yeah. So I think we can jump to the third question, Heléna.
Yep.
Which is how do scientists classify these fossils, and how do they fit into the modern tree of life, or are they something entirely unique?
Right. This is a very fun question.
Right.
And you kind of touched on it in actually the previous discussion about them being a stem group eumetazoan. So the idea is that eumetazoans, being these complex animals, early on, they branched off as this stem, and they went extinct as everything else kept evolving. So again, discontinued. So it's not like, Fractofusus kept evolving and now it's an elephant or whatever. Like, it got cut off and everything else kept evolving. The way that we actually figure this out is this field called phylogeny, which I am growing accustomed with. It is very complicated, but it is fun. And it's lots of computer work, lots of statistics, algorithms, and coding. And it's basically figuring out, if you have these fossils or even living animals as well, where do they fit into the tree of life of what we've previously figured out, just how things are related to each other. So that's basically that.
Right. Yeah. So, for example, I would add that within eukaryotes, like, which is this huge group of organisms, there are some what they call like eukaryotic supergroups. One of them is called the Uniconta, which includes the group Opisthoconts, which that includes fungi and animals, right? The metazoan. Right. So that's where they would be located, as we said, likely as a stem group, eumetazoans, like the first eumetazoans, basically after sponges, basically, right? Something similar to the Cnidarians, which include corals and jellyfish and so on. So I guess what defines animals in general are different things that could be a bit tricky regarding our organisms, because they should be capable of moving at least during some stage of their development.
Yep.
Which maybe we don't have the evidence of. But they will also require the presence of spicules or collagenous-based colonies. Likely sexual reproduction as well, which there's some evidence about that, or some like well-grounded guesses, I would guess. And they also require some tissue differentiation, right, as opposed to the sponges, and multicellularity as well, right? And in terms of eumetazoans, for them to be considered eumetazoans, they should include the existence of true tissues, which we seem to have the existence of true tissues. Some parts of the organisms are preserved differently, and that's likely driven by different consistencies, right? Some tissues being more robust than others, for example. And they should have maybe the presence of nervous systems and muscles as well, which, we have evidence of muscles in some cases. And they all show something that we call a gastrovascular cavity. Whether our organisms have a gastrovascular cavity or not is debatable, I guess. There are some models of them maybe showing something like gastrovascular cavities. It could be. But at the beginning, during the 60s and the 70s, all Ediacarans basically were grouped under like this huge group called the Vendobionta, and then eventually they are being split up into basically different groups and clades and so on. But I think that's what I would say about this. So I guess we can now jump on to the next question. Okay, well, that was it now for phylogeny, your favorite topic, Heléna.
Yeah, thank God.
And now we're going to talk about, I believe this is the fourth question, So the fossils at Discovery Geopark offer a glimpse into ancient ecosystems. What do they tell us about how these organisms lived and interacted with their environment?
Right. So we actually use a mix of the fossils themselves and their morphology in combination with the bedrock to kind of infer life mode, I would say. We know that these organisms lived at the bottom of the seafloor, as we touched on. Some lived upright and some lived reclined in the sediment, and some had these basal discs that we were talking about, and some don't. And that the presence of the basal disc can help us infer whether or not they lived upright, but it's not a sure thing. You can't assume that something lived upright because it had a basal disc, which is very tempting, but not sure. Another thing we can do is actually more related to what you did in your work, actually, is using the bedrock and the paleocurrent of the bedrock. So essentially, paleocurrent is just what direction the water was traveling at the time of sedimentation, when these organisms were alive and right before they were buried. But so that paleocurrent, in conjunction with the orientation of the organisms, you can kind of study that to see how they like to live. And so, like with your research, you studied fructifusis and looked at the orientation of the long axis relative to the paleocurrent. And correct me if I'm wrong, but you found that they had a preference to be in line with the current, right?
Oblique to the current, kind of like
oblique to the current.
Not completely parallel, but kind of like if the current is going like this, just at an angle from the current, like 35 degrees, something like that.
Yeah, so stuff like that, that's really helpful to know, and it can help us figure out how they lived back then. So, yeah.
Yeah, I would say that in terms of how they interact with each other, basically what is done is, I guess, spatial statistics, just complex mathematical methods to basically show if there is competence, or if they have nothing to do with each other, or if they have like some niche speciation or whatever, and so on. And in terms of how they interact with the currents, I think it's kind of like what you said, basically identifying in what direction the paleocurrent was going, which in our case is sometimes it comes in the form of what we call climbing current ripples, which are just kind of like these wavy, just sand ripples, which tell you in which direction the paleocurrent was going. And basically, if you had organisms that were living upright, erect into the water column, and you know which way the current is going, you would expect to see all the organisms failed and aligned
following the same direction,
right? If that is not the case, which is in many cases not the case, it's likely there's something else going on. So I think this is one thing, the paleocurrent direction, and the other thing is, as you said, preservation. For example, sometimes when we have organisms with a stem, you have beautiful preservation of the holdfast or of the basal disc, but not really the stem. And this is sometimes because they get curved, arched up away from the sediment, and there's just
like that.
Exactly, and there's accumulation of sediments, so they get preserved at a higher plane that we cannot really see, right?
Yeah, exactly.
For example, sometimes I've seen organisms that have a set of branches basically folded under the stem, and that could have only happened if these organisms were upright, because if they were reclining, it doesn't seem very likely that the branches could have been failed and like folded under the actual body of the organism. So I think that we combine these two things, the preservation and the hydrodynamics, I guess, and the sedimentation.
Yeah, really.
Okay, so we're gonna keep going now. This is the fifth question. So this period is often seen as a precursor to the Cambrian explosion. How do these fossils help us understand the transition to more complex animal-like life?
Yeah, so I guess really studying the Ediacaran period is so important because it's the starting point for everything that came after it. I mean, I'm biased, so it's the center for me. But the Cambrian explosion is all the new things that we see in the Cambrian explosion are only significant if you know exactly the starting point of the Ediacaran. So, for example, in the Ediacaran, there's not really predation yet, so everything is soft-bodied. So in the Cambrian, when we start to see like skeletons and chitin and all these hard parts, that's really significant because we've studied the Ediacaran period enough to know that didn't exist then. Right. So yeah, and like similarly, like locomotion, we don't see a lot of it in the Ediacaran, / none, very little. And we see a lot of trace fossils all of a sudden in the Cambrian. And that's only significant because we know that there weren't as many in the Ediacaran period. So in terms of just like trophic networks and understanding the whole cycle of that and the hierarchy, it's all relative to the Ediacaran. So it's important to know where we started.
Yeah, I think it's important to understand, as you said, like the trophic development, the ecological interactions, and so on. In terms of movement, for example, that you mentioned locomotion, it's true that we don't have a lot of evidence for movement. So the Ediacaran is divided into 3 assemblages, basically. Our assemblage, which is the Avalon assemblage. Then you have the White Sea, and then you have the Nama assemblage. So it's true that in younger Ediacaran assemblages, there is some evidence for locomotion. It's not really our case, apart from, I guess, Hautia, which had muscles, so it must have been able to move.
Yeah.
So and in terms of like how it is related to the Cambrian, there are several hypotheses of why is it that a lot of Ediacaran organisms, virtually all of them, disappear into the Cambrian. So it's not really very clear if there is like maybe like a mass extinction event that basically wiped out all the Ediacarans, or maybe like an ecological thing. Maybe they were like outcompeted by Cambrian competitors that had evolutionary innovations that made them more fit than way more fit. Exactly. And other people also talk about the closing of a taphonomic window. Basically, as we discussed earlier.
Oh, interesting.
For Ediacarans to be preserved, you need them to be sealed by sediments, then sealed by microbial backgrounds, which keep them basically under anoxic circumstances where sulfur-reducing bacteria can precipitate pyrite and so on. But the thing is, into the Cambrian, you start having bioturbation, so little organisms that dig and disrupt the sealing microbes.
Yeah, it's like everything kind of gets, like, the sediment goes up and there's no hope for soft-bodied organisms to be preserved.
So exactly, you have like these organisms digging, disrupting the microbial background, so there's probably some degree of oxygenation and so on. So the chemical process by which Ediacarans get preserved during the Ediacaran is less likely to take place in the Cambrian. So maybe they were there, but they were not preserved. There's a hypothesis that is called the Cheshire Cat from Alice in Wonderland.
Okay.
And it's just
that's fun.
It's a fun name. But so it's and there's some alleged Cambrian survivors. So some organisms that. What is that? Did you see all the balloons in my screen? Normally I cut these things from the video, but this I'm gonna leave it. I liked it. But so there's some organisms into the Cambrian that are like alleged survivors from the Ediacaran. One of them is called Stromatophares, and another one that is called Thabtentilum or something like that. I can't remember. It's published by Simon Conway Morris, and so it's possible that some of them survived into the Cambrian. There are some examples.
Yeah.
Okay, so we can jump on to the next question, which is question number 6. These organisms are very different from modern animals. How did these organisms feed and reproduce?
Right. So I feel like we've touched on this, but because they're soft-bodied, there's very little to go off of. So everything that we say about this is heavy speculation. So we think that they fed through either like filter feeding or osmotrophy or some sort of very passive form of feeding. It was, again, no predation. There was no that's how we think they fed, but it's still not certain because we don't have any like internal preservation of the organs.
Right.
And in terms of reproduction, as far as I can tell, we really don't know. I think both sexual and asexual has been proposed, but you had mentioned that there's some maybe more evidence for sexual reproduction, but like we really don't know.
Well, so I mean, I guess what I'm going to add is in terms of feeding, it's true that the main two theories are one, suspension feeding, but there's some problems with that because we don't have any like feeding polyps or anything like corals preserved. We don't really have pores, so they could just be like somehow a passive suspension feeder with all the particular organic matter falling out of suspension just gravitationally. So we don't have evidence of these structures. In terms of osmotrophy, basically that involves the external secretion of enzymes and then the ingestion of the result of the external digestion. And there's some problems with this because we don't believe that the pool of dissolved organic carbon in the Ediacaran oceans was large enough to sustain these organisms, at least not in an obligate manner, maybe like facultatively as well. So using osmotrophy at the same time as using other strategies, right? They were not like fractal enough to have a surface area to volume ratio that was enough to conduct solely osmotrophy, basically.
Yeah, and not all of them were fractal too.
And not all of them were fractal, yes.
Like, yeah.
And
the ones I'm studying, Primo candelabrum, that's not fractal.
Right. And some of them were even in touch with the microbial background. So some of them have like their ventral surface, not only the dorsal, also fractally branched in close contact with the sediment. And that's interesting because the sediment would have been exposed to high concentrations of very reduced forms of sulfur, like sulfides, which are toxic for cellular respiration. So they must have had a way to cope with these high concentrations of sulfur, and we believe they could have been establishing symbiotic relationships with sulfur oxidizing bacteria that could have basically detoxified sulfidic pore waters, basically.
And I know that like the basal discs are sort of their role in that speculation is also on the table as well.
Yeah, they were in touch with the microbial background, so they probably had something to do with that. And in terms of reproduction, I mean, it's true that we have juveniles that are identical to the adults in a lot of cases. It kind of like points towards something close to asexual reproduction. We have observed some structures that resemble some like budding structures, kind of like asexual budding. In terms of discs, we have some discs that are kind of like merged together. They're called kissing discs, which could be like asexual reproduction as well. Some filaments, like stolon-like reproduction, connecting organisms, but this is tricky because these filaments are not preserved everywhere, and they also sometimes connect different species as well. So they wouldn't really be asexual. Some authors have hypothesized that they could have reproduced by waterborne propagules, either sexual or asexual, and stolons. It's true that given their global distribution, they must have had a way to reproduce by some waterborne structures, either sexual or asexual, that allow transport basically all over the globe, really. But it wouldn't be unprecedented if they combine asexual and sexual reproduction strategies. This is common in eukaryotes, so it's a possibility. Okay, so I guess we can jump on the next question. This will be question 7. So why is Discovery Geopark such an important site for studying the Ediacaran, and how does it compare to other fossil sites around the globe?
Okay, right. So the geopark, and I'm also just going to say Newfoundland in general, is like exceptional for Ediacaran preservation. In like, if we just look at Canada, it's basically the only place you can study Ediacaran fossils. There are some in Northwest Territories, but they're not as well preserved and not nearly as numerous or dense. So it's really, really special. In addition to that, having the preservation that we do is just, it's a miracle, honestly. Having that tuff layer that we were talking about, and you can actually see it in some of the surfaces. It's still there in parts. Having that layer preserve it is really good for the fine detail. So it's really a really good site for having finer detail preservation that way. It's also incredible because it's so old, as we talked about. It's unusual that hasn't been subducted or anything like that. So that's really neat as well. And then the other thing is, Danny, you mentioned this already, but the Ediacaran assemblages, there are 3 of them, and the ones that are found in Newfoundland and the Geopark are named after the Avalon Peninsula, actually. So it's the Avalonian, and that's the oldest of the 3 assemblages. So not that we're better, but our Ediacaran fossils are the oldest of the 3. For example, the ones in Australia, which actually Ediacaran is named after the Ediacara Hills in Australia, those are younger than the ones that we find here. So
they are, yeah.
So it's really, really special that way as well, I'd say.
Yeah. So in terms of the Avalon assemblage, there's also fossils in Charnwood Forest in the UK. They look very, very good as well. And as you mentioned, there's beautifully preserved fossils as well in the other 2 younger Ediacaran assemblages. For example, in the White Sea assemblage, which includes fossils from typically from Russia and Australia. Yeah, they're beautifully preserved, very different from what we have here.
Very different.
And the preservation is also mediated by microbes, essentially. They don't involve the presence of ash, really, like we do. So it's more unified by the action of microbes. But those are also beautifully preserved. I have to say that I have never been to these sites, so I haven't.
Yes, me neither.
I haven't witnessed this with my own eyes. So I'm just speaking in terms of what I've seen in scientific publications, really. Okay, so Heléna, I think this is going to be the last question. Looking ahead, what are some of the biggest questions that remain about the Ediacaran fossils, and what future discoveries could change our understanding of this ancient world?
Right. So I think in general, classification is a really big one, both internally within the Ediacaran biota and externally, sort of relative to the rest of the tree of life, as we were talking about. So increasing the number of studies of Ediacaran phylogeny would be really great if we could resolve some of those groups, just because they're constantly evolving. That really threw me for a loop when I first started studying this, because I thought things would be set in stone as I learned in school. And then I got here and it was like, oh, well, that changed in this year. And there's this new discovery that showed this. So it's constantly evolving, and it makes things really hard to classify in this framework that is always shifting. So it would be really, really great to have a more sort of permanent framework for that. We need to know how they relate to the rest of the tree of life. There are lots of different interpretations, and lots of work has been done on it, but all of them are slightly different. There's not really one consensus that we know exactly what's going on. So it would be nice to get some clarity there as well. And I think just in general, we need, in terms of what discovery could help us and would be the most beneficial for us, would just be to have better preservation. I mean, not to be greedy, but we just need more. We have good preservation and it's awesome, but we need even more to know these things. We need the internal anatomy preserved in order to be sure about things being related, because we're sort of using very surface and external features to relate those things at this point. So yeah, that's my take.
Yeah, there's some internal preservation in younger assemblages. I'm not very familiar with it, but I think there is some internal preservation of internal structures.
Yeah.
And I'm not sure what else to add, really. I agree that taxonomy, basically classifying the fossils, is kind of like the foundation of everything else. Without good taxonomic studies, it would be tricky to conduct, for example, ecological studies, because if you don't really know what kind of creatures you have or categories you're working with, maybe it's possible that you conduct Ediacaran ecological studies that become quickly outdated by newly evolving taxonomic studies. So I think that is the solid base of everything else, kind of laying the groundwork, if you want to put it like that. And yeah, I think it's in the future. For example, I've been working with something that is called computational fluid dynamics lately, and there's basically consistent computationally modeling flows, kind of like to recreate the flows that these organisms experienced in life. And there's always new models, new technology, like supercomputers that can do it a lot better, model the behavior of single particles, which is being done now. So it's interesting. There's always new technology that will help us to just look into this a little bit farther. And well, I think I don't have anything else to add. Heléna, what about you?
Yeah, no, I don't think so. Yeah, I mean, for me, in terms of my work, I feel like the biggest question for me, which is a very niche part, is the species Candelabrum. We have some at the Geopark, actually, that look very different from the ones that are found in the UK, and that kind of affects the classification as well, and whether or not they're rangiomorphs. That would be really nice to resolve as well. So maybe some new discoveries of Prima Candelabrum and sort of the branching would help unify that a little bit better. But yeah, I think it all just goes back to differences in classification. They just need sorting.
Right. Yeah. The holotype of Prima Candelabrum is in
yes,
Discovery.
It is.
Yeah. And,
yes, it is. Very famous.
Yeah. It's actually, when we were talking about iconic Ediacaran fossils, this one is probably not very iconic, but I always liked it. Next to the holotype of Prima Candelabrum, there is this guy called Hadroniscala, which is like this ladder-like.
Exactly. So cool.
Yeah, ladder-like enigmatic
that
fossil. Nobody really knows what it is, but it's actually very cool. Okay, Heléna, well, thank you for participating, and I hope you enjoyed it. And,
yeah, it was super fun.
Yeah, and I'll see you all in the next podcast, and that was it. Bye-bye.